right? Everything's good?
Yeah, so. Got it. So we're really happy today to welcome CarolynLarabell is our seminar speaker, and she's going to talk to us about X-ray tomography. So Carolyn is a professor of anatomy. She's in the School of Medicine at UCSF, and she you can correct anything I have said here wrong. For SACS at the Advanced Light Source at Berkeley until I don't know what was through NIH's mind. They decided to close the facility, and so she's moving her instrument over to the accelerator at Stanford. Biohubs of California are making a major contribution to get it up and running. And so I think you'll see from her talk why it is so important for us is that they always wanted us to give a little bit of an overview why this is how does what we present here fit in with our missions of our center, which I explained to Carolyn is to make 4D, and being able to image an entire cell is really important. And when we say cells, we started off with a minimal bacterial cell. We're moving on now to a yeast cell. With I think anybody who has published on this, she's listed in the author list. And just to give you a little bit of a comparison, because I know you've seen some of the tomograms that we've had from Julia Muhammad about FibSim. But if you look at the spatial resolution, yeah, it'd be great if we had a cryo-ET of the whole cell, because you know we went 2 to 500 nanometers. But with the SACS, you see everything. It's about a 35 nanometer resolution. And if you do FibSims, which is multiple slices, you lose some of the accuracy that you had with the ET, but also look at the times it takes. 10 minutes for the cryo-ET, but we'd have to ask Julia's postdoc what it really takes her to do, and they do the FibSim, that can be days to weeks. And also get a little bit of an idea about the thickness of the sample that you can handle. So the soft X-ray tomography is really in that sweet spot where we need to get a picture. And I think outer window, they like to work, but I did learn from Carolyn this morning, not every accelerator, some of them can have too powerful of a source and is not really designed to connect well into their instrument. And this is from a paper that they have in bioarchives with Wallace Marshall. And I think most of you know he is a director of a center out at UCSF. And these are yeast cells. And I know so Charlie always quizzes people, you know, which one of those objects in the cell is the nucleus? And it's not the biggest object, it's the smaller one. And you see the vacuole, and those are lipid droplets. But in this wonderful dividing, and please notice you're being able to see the mitochondria network there. And I also, Wallace arranged that we got, I think, about 100 different tomograms from them, from the wild type yeast. One other work that was done by the group of Chang, he's in Taiwan, also on yeast, where they were able to even have a better work and how it changes at different times of the cell cycle. So this is just unbelievably rich information to start a host cell model, and you know, of course, and then start calculating some biological system like a galactose switch, and you could be following this because you have such good resolution. Electron tomography, which gives you the interfaces, you know, beautifully. The thing that I noticed that it's difficult for all of the methods to give you is information on the endoplasmal reticulum, and that is something that Julia Muhammad is really working on with her tomograms at the moment. And also, you notice in some of the yeast pictures, you don't always see the mitochondria cannot be automated, so you don't always get that, but you will get the droplets and the nucleus and the vacuole. So this is sort of the state of the. And so without any further comments, let's welcome Carolyn for her talk. And I know we're going to learn a lot today.
Thank you very much. And it's really a great pleasure to be here. I look forward to this for quite a while. We've been trying to work on making it happen, and it finally happened. So that's great. This is just a picture of a nucleus with the various heterochromatin and euchromatin filaments in it that we segmented, but I'll move on from there. So the features, I don't have to pull cells in the hydrated state. I'm gonna move this down here so I can face you and do this at the same time. They're cryomobilized, just like for cryo-ET. That's because you have to mitigate radiation damage. And also with tomography, things move and you want air to. We get a natural contrast, and that's from imaging in that water window. And I'll tell you more about that. 25 to 60 nanometer resolution. I'll tell you why, the range, and what we haven't talked about yet is that to get molecular information on top of the structural information. So first I want to give you a feel for the vast types of cells that we can image, because everything with carbon and nitrogen can be imaged, and we've imaged a lot of different things. Seeing before budding yeast, and this one we have segmented mitochondria. You can see the little tubes, and we've looked at the other yeast, the pombe yeast, and you see the nucleus, the mitochondria, and vacuoles appear. In this one, you see smaller vacuoles than in the budding yeast. The budding yeast. What we've learned with imaging a lot of different people's yeast cells is they vary. There's a lot of heterogeneity in the way a yeast structure looks, depending on whose lab it's grown in. We've looked at algae, and this is a multicellular algae. It sort of started as a single cell and divides into multiple cells. We can look at spores, so even going through a T cell, this is one of the blood cells. You can see the mitochondria and ER that you see, and they're not ER in this one, sorry, nucleus and mitochondria and some lipid droplets. We've looked at the nucleolus of a neutral nucleus because they're designed to squeeze through small capillaries and things, so they have to be flexible. Chromatin organization in an olfactory neuroepithelial cell. The nucleolus of a eukaryotic cell, and the silent X chromosome, which I'll talk more about, is popping up right there, labeled with a GFP tag. And candida, you can get the long candida growing in these capillaries. We can also get bacteria growing in colonies in the capillaries, so they're already in there. We managed to look at both the surface view and an absorption view, where you see the very dense heterochromatin in the sperm head. So I mean, a vast variety of cells that you can image with this technique, the details. But two unique elements. One is the light source, where we use X-rays. So you can't use iridescent light or light bulb or ion plates. And the contrast mechanism is the absorption of X-rays, differential absorption of X-rays. And I'll get into all of this in more detail as we go along. Just a view of the CAD drawing at the top shows you the important things: the bin magnet from the synchrotron, safety valves. The only thing a user sees is in the yellow square. That's the in station, and that's all the user ends up seeing. The rest of it's behind shield walls, etc. But our station does not have to be behind shield walls. Our X-rays are low energy, soft X-rays. That's what the soft means. So you don't have to. The specimen stage we use a full rotation goniometer, and that means you get isotropic resolution. There's no missing wedge. And so it has rotary motor at the top, and the specimen hangs down into the X-ray beam and full rotation. The other thing is it's at atmospheric pressure. All parts of the microscope, except where the specimen is in a vacuum. So what we do is run a stream of liquid nitrogen cooled helium gas, cold helium gas, across the specimen very gently throughout imaging to biose all ice off the specimen. So you don't get any ice formation throughout the imaging process. And we have to use a helium gas because we are imaging that water window. The nitrogen would absorb. So it's a helium gas. Image formation. Very simple microscope. Really ignore all the other complex parts. It's just the specimen, and the objective zone plate magnifies it and stores the image onto a CCD camera, since it's imaged onto the CCD camera. So it's really very simple layout for a microscope. The nanofabricated device, and the way they work is those rings get narrower and narrower as you go out to the rim of the zone plate. And that width of that outermost zone is what limits the resolution, or determine the aspect ratio of that outer ring is the efficiency of the zone plate. So as you might imagine, as you get a better resolution optic, you get a decreased depth of field. So we want a better depth of field in imaging to 15 to sometimes 20 microns using some tricks that I can't show today, just due to time, but occasionally you can get a 20 micron. The optics are what's limiting our resolution. Our wavelength is 2.4 nanometers. So why can't we get to 2.4 nanometers? These optics are so difficult to make. Getting a 10 nanometer resolution optic is only done. The really cool thing, though, about the technique is that contrast mechanism in the water window. Water window being that region of the energy bandwidth between 284 and 543. We image at about 520 eV, 517 EV. And in that range, the carbon and nitrogen in a specimen absorbs the X-rays an order of magnitude more than the water. So unlike cryo-ET, where ice obstructs your vision of your structures, ice is invisible to us. So we're below the oxygen edge. We don't see the water, just mostly carbon. And the cool thing is it's linear with thickness and concentration. So the higher the concentration of carbon per voxel, the greater the contrast. That's the thickness and concentration. So you can get a distinct linear absorption coefficient measurement for different structures. So like in this cell here, you can see the nucleus, lipid drops, ER. This is one of the cells where you can see ER, and mitochondria. Mitochondria have double membranes and highly absorbing in this tool. ER are not as filled with carbon, and a little lower contrast. And you can see euchromatin in the center of the image. I can put this little red thing so people on Zoom can see it. And these dense blobs of heterochromatin. And then the nuclear envelope with heterochromatin just beneath it. And this reflects the linear absorption coefficient value for each of those regions. So as you can see, carbon lipid droplets were very highly carbon filled are the highest absorbing and the highest contrast. We have a specimen in its normal growth medium. We don't do anything but take it from that normal growth medium. We make a pellet. I take the specimen from that growth medium, put it into a glass capillary, and then rapidly freeze it. And we have an automated cryo puck in there that the well down there is filled with liquid nitrogen, and you can hold capillaries as they're frozen. So you can freeze a lot of specimens at one time. And that's the actual device in the lab. A lot of are warning people, keep your paws away. So that's what the flashing lights are all about. Once you do that, you take the cryo-frozen specimen down to the microscope. You can take your whole puck down and liquid nitrogen, let it sit there. Put the microscope 90 to 360 images all around the specimen, just rotating. The larger the cell, the shallower the rotation, 1 degree around the cell. And then it's automatically sent up to the computer and automatically aligned and reconstructed. So that whole reconstructed cell in front of you at the microscope. And you can see that's some sort of fibroblastic type cell. I can't remember which one that was, but you can see all the different structures in there. There was an array of ER on the bottom of that. Look at the array of ER at the very bottom of the cell. A lot of high contrast heterochromatin in the center of the nucleus. Now, because it's so fast, Nady, and I'm going to show you another cell from that exact same dish that looks very different.
Can I ask a quick question?
Sure.
So like with the yeast, when I asked you, well, like, why would there more mitochondria in your like bioarchive? And you said, well, because that has to be done by hand.
Yeah.
The others are automated. But here you can.
Implementation of the mitochondria. Yes, that's what a qualifier I was going to have to tell you about.
Oh,
These are harder for some reason.
so but in human cells, it's oftentimes easier.
The problem there is you can see them, but they frequently contact other organelles. You have to have
Okay.
decision making, but it is much easier. Okay, so there's a lot of, you can do a lot of stuff in a short period of time. Now, why capillaries? Why not a flat grid, or a flat piece of glass like the light microscopy? They're limited to the, yes.
So I can ask a question very quickly about the microscope setup on the previous slide. So what is so specific about the source of the soft X-ray there? Because the rest of the setup seems
Synchrotron.
like that's
There's now a tabletop that's being built in Ireland, and they're trying very hard to sell one.
Yeah.
It's flux limited, so it's going to take much longer to collect a data set. And they're optimistic, but it really takes about an hour. It takes about an hour to collect a whole cell.
I see.
Flux
Does
limited.
it give a similar resolution and everything else?
They
Or-
claim yes, but I haven't
Yeah.
seen it yet.
Thank you.
So to solve the limited tilt problem, we use a full rotation. What does that really mean? You look at the bottom here, you can see with limited tilt, you've got missing information that you don't have in the specimen itself. Gets thicker and thicker, and there's more obstruction from the specimen itself. It also causes more radiation damage because you have to use a higher dose of X-rays at higher tilt. Biggest problem is, you've limited your abilities. You've got an X-ray microscope that can image up to 20 microns. Why not do it? This limits you to a 5 micron thick using a flat specimen. So what do we do? And also cells in vivo in our bodies aren't really flat either. They don't grow on ground, they grow in tissue. So what we have done to try and make all worlds possible to limit adherent cells, we made this device where these are hollow tubes that are magnetically attached to this device back here. You can see the red arrows pointing to a capillary that's going down that tube, and the tip goes down into the fluid in a 96 well plate. The incubator, let them grow and adhere to the capillary. You can do experiments, change the medium in the 96 well plate, and conduct experiments while they're in these capillaries. And then you get images that look like this. You get a whole row of cells up the capillary. And that actually looks like a fibroblast you would see. It doesn't solve the problem of people who have cells on a grid that they want to image with the X-rays. So we're working on making it possible to do flat grids, opening up the space between the optics. Segmentation, our manual segmentation and automated is assisted by that linear absorption coefficient. And so I'll show you some of that. We're looking at the whole cell. You first segment the nucleus. So you round it off, isolated the nucleus wall. And so you can put all those voxels up on the computer. And you've done that by looking at the nucleus and tracing. Once you've got that, you can look at the program online. I mean, on your computer, these are identified as heterochromatin based on old EM pictures. And you know that's the heterochromatin. So you adjust your computer to grab all those voxels in that range, just above that specific linear absorption coefficient range. And so then you can case all of them. As you see here, they're all isolated now in our Amira program of the heterochromatin and euchromatin in the cell. So dark is the heterochromatin, and euchromatin is the low absorbing region. And then you see the 3D distribution of the heterochromatin and euchromatin throughout the cell. Your mitochondrial network in a human cell. And you see how, and it's really a beautiful network of mitochondria. You can now get volumetric measurements and spatial distribution measurements, et cetera.
Is this in the log?
Exact phase it's in.
Okay.
This one is not post-mitotic. That one I do know. It's pre-mitotic. It's from tissue. Yes.
So amazing images. I was curious whether it's possible to look at the DNA within the mitochondria.
About that later because there are things I'd like to try to be able to do that, like labeling with BRDU, and see if you
Yeah.
can get a contrast enhancement with BRDU or something like that. We haven't, I need somebody, and then we can wrap that mitochondria around the nucleus, so you can tell how it's interacting with the nucleus. So you can build the cell with all the different components in it. And we calibrated our linear absorption coefficient by structure of the alcohol oxidase has been done with cryo-EM. So we could calculate the expected linear absorption coefficient based on composition of atoms in a voxel. And it came out to exactly what we collected on our X-ray microscope. So it's a valid measurement for the molecular content of the cell. And as it changes, you can detect changes in that. So visualizing nuclear structure, protein condensation during blood cell formation, hematopoiesis. And these are all cells that were obtained from mice, and they had markers to know which stages they got them from bone marrow and then the circulating cells. Starting at the top with an embryonic stem cell, some progenitor cells moving down to the fully differentiated B cell and neutrophil down here. And what you could do then is calculate the ratio of heterochromatin C. The percent heterochromatin increases tremendously as the cell differentiates and genes are turned off. Now remember, it's percent heterochromatin. The volume of the nucleus is decreasing. And so heterochromatin content itself isn't really increasing that much. It's just that the volume of the nucleus is decreasing. And so you get a higher percent heterochromatin. On mammalian cells, and it's exact inverse when you have go from a normal to a cancer cell. You go from a small cell to a humongous non-differentiated cell in a cancer cell. The other thing we can do is look at the thick band of heterochromatin that forms around the periphery, the amount the thickness of that band during differentiation, and you can prove quantitatively that the thickness increases in differentiated cells. It's not just a visual technique, we're getting quantitative validation of our visual observations. We did a very similar test on neurogenesis, olfactory epithelial neuron differentiation, again from mice. And this is olfactory receptor gene selection story by Stavros Lombardis lab. And it really in a cluster across 18 mouse chromosomes in every cell in the olfactory epithelium, 1,500 genes. But each of those neurons expresses just one allele differentiation. And so that was what Stavros was interested in studying. And so we got, again, markers that would differentiate between these various stages, epithelial cells, maturing, and stem cells. Got the various markers. So we dissociating tissue. And so you had to have markers for which cells you wanted. We were able to do that. And you can see, again, like the hematopoiesis, there's a change throughout differentiation, but it's the exact opposite of what you see in also retinal cells. But you can see the heterochromatin leaves the periphery and moves to this central mass in the fully differentiated cell. You can quantify those changes, the percent heterochromatin, 33%, 1%, and then you can do chromatin with respect to the nuclear envelope, as you can see visually, as well as in these plots. And what Stavros noticed with these studies is that these 5 or 6 of these foci around that central mass. And so he focused in on those as being perhaps the clusters of silenced genes that are formed during the differentiation stage. And use the same cell, he showed that indeed those are the silenced genes. One gene pops out during differentiation, and that's the one that would be expressed. He also noticed that during that last lamin B leaves these cells, the differentiating cells, and the mature cells have no lamin B in them. So what he did was force a mouse to express lamin B, and you disrupt that central localization of chromatin moves to the inside. And if you force the lamin B to stay in the nucleus, you also disrupt gene selection. So that 3D structure organization is critically important for proper genes, because if you knock out HP1 beta, you also lose that localization, which we can see both visually and quantitatively here. So this is a case where the 3 is critical for gene expression. He later did an experiment where he started culturing them on flat substrates, and he blocked proper gene selection because everything disappeared. Quickly going into some environmental application experiments. You have the single cell yeast top and 16 cell yeast on the bottom. I'm sorry, not yeast, algae, microalgae. And you can look at the different structures within nucleus in purple, mitochondria red, and starch in this outer bluish color. With glucose, and after 84 hours of glucose, there was a huge increase in the production of lipids, which they were able to quantitate. And not only in production of lipids, but also they were around the whole algae after about 84 hours. And it showed there was a 5-fold increase in starch and a 16-fold increase in lipids. So you can do experiments where you can quantify changes in components of the cells. Specific mutant on the DNA, they don't have a difference between heterochromatin and euchromatin. They were looking at this supercoiled domain and looking at the 2 different regions there. And when you switch from an HUAA to HUB, you flip the lac evaluation to, and in the stationary phase, it's all lac 0.29. And the bacteria itself is shrunk in length. So this is the nucleoid of bacteria than you would get from other technologies. And again, they looked at other treatments of these bacteria and combined it with Sachs measurement to look at the structural change that occurs in that particular molecule, small angle scattering. And this is one of my most recent and most fun studies was, is it an endosymbiont or an organelle? This is our recent paper we did with John Zare, and he, Tyler, Kendra was a former postdoc, now assistant professor, and Tyler was a postdoc who did a lot of the work. And their question was about this Bigelowi and UCYNA, which he calls UCNA, cyanobacter. And the cyanobacter moved into the algae beyond, and he had data suggesting it had become an organelle. So he wanted to do the experiments to determine that. One, he showed that many proteins, and you could see that they were encoded by the nucleus, expressed, and then transferred over to the to the UCNA. He did some evolved, those in UCNA and those missing in UCNA, and those that are imported from the cytoplasm. And so many additional functions are provided by the host to the mutant, the endosymbiont UCNA, and 50 functions from a Bigelowi. The genetic modifications. Question was, is it synchronized? Is this cell cycle independent or is it coordinated? And so that's when he turned to us to do the DNA and how do they grow and divide? How do they live together? So they have a dark cycle and a light cycle. Light cycle during the day is when they photosynthesize, and dark cycle at night is when they undergo cell cycle changes. We've got this beautiful mitochondrial network in green wrapped around the UCNA, the endosymbiont, in aqua color, cyan color, I guess it would be. And then the plastids in the purplish color on both sides. The mitochondria is perfectly positioned in those cells to give the endosymbiont some mitochondrial energy, some ATP. And we could quantitate the volume of each of those structures and the lac values of each of them to see if there were any changes during the cell cycle. We then looked at various time points, and what we noticed that the very first change that we could see was in the mitochondria. Mitochondria doubled in volume. You wouldn't be able to tell that by looking at it, but because we can segment them and determine them, and that was the very first thing that would happen. And then we would see that the UCNA was starting to replicate and divide. It divided, and the DNA in the nucleus was replicating and starting to divide, and plastids were replicating and starting to divide. And then you can see you have 4 plastids now, 2 UCNA, 2 nuclei, and double the mitochondria. After that mitochondria doubled, I should point out separate networks, one on either side of the nucleus. And then you could see that they separated into 2 completely new organisms, highly synchronized. And again, the quantitative data to support that the doubling of the volume of mitochondria changes in lac values. It's a visual study to justify the volume of the mitochondria increased first. So that sort of cinched it, that we decided that it had evolved into an organelle. The reviewers encouraged us to be. Magazine chose it's one of the top 10 breakthroughs of the year in 24, which was exciting. The paper was published in Science, which was exciting. And then we just into February got the Newcomb Cleveland Award, which is the year. So even though NIH doesn't like what we do, I feel vindicated. I think we've proven that it can do exciting science. So that quickly, because I think I'm probably pushing time. I have no idea what the time is.
It's about 13 minutes.
Okay, we'll go quickly. Sickle cell anemia, you need to look at large numbers of cells. And what we could see is that this was a study to look at the effects of drugs on reversing sickle cells. And what you couldn't see in the hemoglobin fibers, and what our hypothesis was wrong in the end. Turns out the more of these little projections you have, the younger the cell is, and that spiky ends. But that shows the difference from a top view and a side view, the value of 3D. And that helped them to have a new tool to evaluate their drugs. We also looked at SARS-CoV-2, the lab. So we did that, and you could see the formation of the condensed membranes in orange. A lot of the cells have a double nucleus. They fuse, they join another cell and fuse together. And DMVs, I don't see as many DMVs on these. There you see the DMV. And virion entries, you can see where the actual virus particle was entering and the condensed membranes here. We've also looked at herpes simplex. You can see virus particles on the surface of the cell as well as in the nucleus. And you can quantify them, tell you how many particles are on the surface, how many are in the nucleus. And you can differentiate them by presence of a capsid. Presence of capsid gives them, and the presence of the envelope around the virus gives them a higher lac value. So you can differentiate between the virus particles just based on lac, based on the lac value. Again, just looking at the formation of the structures that viruses form, these elaborate membranous structures that apparently transport the virus particles. Pancreatic beta cells, where we're looking at in quantitative study, again, you can count the number of insulin granules. They have specific lac values that were determined. And you can look at the effects of a drug on the secreted insulin. And so this is a nice assay. You can actually quantify it. And what it means is you can identify the pancreatic. You don't have to have a labeled mouse model. You can do it in humans. So you can get pancreas from human tissue and study the insulin granules without any labels. And we did a correlated study. This was done by Grant Jensen's lab, where we did the whole cell study, and they showed the presence of a crystal in UET and looked at what they called communities or neighborhoods in the cell. So, they're compatible. You get the whole cell quantitative information about insulin granules, or you can see the structure of the insulin granules themselves by putting the two tools together. Now, people at Scripps, he took our insulin granules and the proteomics of all of the molecules that are involved in an insulin granule and modeled how they would have to pack into that insulin granule to give you a crystal values. We now know that when you have this very high absorbing lac value, you've got the crystal in the center, you've got a transitional one with a medium lac value, and the lower to the high resolution imaging, whether or not it's a mature granule or immature granule. That's a beautiful study.
Oh, you mean Goodsell?
Pardon me?
Goodsell, David Goodsell.
David Goodsell and the fellow who, how do you forget Art's name? Art is you. But yeah, that's a David Goodsell and Art. It has a signature. Membranous organelles, we don't need membranes to see organelles. Look at EM, and we can see them based on their lac values. That's the outline of the nucleolus itself. The darker stuff is heterochromatin. You can segment those regions. We did, and you can see the movie. The blue in this one is the heterochromatin. You can see how heterochromatin completely enmeshed in it. And then we thought, logically, the lowest absorption region should be where DNA is transcribed. It has to unwind, and that's where it should be transcribed. So the rDNA label, you can see that is the nucleolin. And the rDNA is in this region, and that dense drop is the nucleolin. The rDNA is indeed in that lowest absorbing region of the cell, and we validated it with a live cell expressing a transcription factor. In these same studies, this was the differentiation of the neurons. The postmitotic, you see these clusters of very highly absorbing particles that we are hypothesizing might be RNA transcripts, but we don't have any direct evidence. But logically, it makes sense to us, and someday we'll be able to find somebody who has the labeled cells that we can actually prove it. Protein condensates, these are poly A, poly U, poly C, and poly G in vitro. And you can see they formed a phase separated or adhered to the glass wall. But poly G forms a fractal structure. Combine them, the A and C at 50-50 ratio. We did the X-ray imaging, and you can see there's a condensate inside of a condensate, actually, or up against the glass wall. And you combine the A and the C, the G and the C, and you just get a different fractal structure. So we were able to look at membranous organelles that way. And now we're going to look at correlated imaging, because this is how you get some molecular information. I don't have time to go into the light microscope, but we designed a cryo-immersion confocal microscope, so you need immersion lens. And so these are 4 optical sections through the inactive X chromosome. It's labeled with the macro H2A GFP, which preferentially binds the different sections. You can see now you know exactly where it is in the X-ray. Don't even need to do the overlay, the highly absorbing structure. But the overlay does prove that that is the inactive X chromosome. And then you can, and what you can see is there are 3 different degrees of compaction within that inactive X chromosome. 0.34 is the most highly absorbing, that's the darkest, the most highly absorbing structure in the chromatin, in the inactive X. And not all of the nucleoli and structures look the same. That's a little bit different structure. This is yeast, and this shows you the difference in the contrast in yeast. And I don't know why that is, but this one is expressing some molecules that are known to form condensates in yeast, and they're involved with collecting the junk in the cytoplasm, misfolded proteins. And what we were able to show is that they form condensates, nuclear condensates too. Very quick tour through that, but I wanted to get some lac-enabled modeling, because this is what we could do with just using the lac values. And this was a study by Frank L. He had collected all of the high C data of a bunch of B cells that had been done by one study, and was modeling how those chromosomes would have to pack to fit into the nucleus, how long to fit inside of a nucleus. And he came up with 3 different models for how the chromatin would be folded. And he was first focused on the centromere protein and showed that there's a model where you form 3 different clusters of centromere protein and sitting around. And it turns out that he was able to show that these 3 clusters, we were able to see them with the X-ray microscope. Now, this is not the same cell exactly, but he had the GM cell that you can get from the company that they guarantee is diploid when they send it out. And you can see that this matches his model of 3 centromeres in this B cells. This was by a mathematician. He took a protein of arbitrary size and binding site arbitrarily and modeled the path of the molecule through the euchromatin region. Wouldn't move through heterochromatin. Well, the heterochromatin is too dense, but modeled and looked at the median search site. And he found that there's an optimal density of molecules right about here. Too low, they don't travel well, and too high, they don't travel well. That was the lowest travel time. And that's just based on lac values. This one was looking at the site, looking at gliding, and they did a correlative study with live cell actin imaging and the gliding movements these parasites make. And they used our SXT to get the surface of the cell and make a mesh of the formation of the actual structure of the model parasite, and showed that it's a sort of a self-generating self-organization, not a microtubule-based template that was combining. They used our shape of the organization and depolymerization to do that. So that was a kind of nice study too. And then I'll just thank all the people in the lab and show you a day at the NCXT of people imaging. Mark Legros in the upper left corner up here is the physicist who designed and built the X. She's now in Germany. Jianhua Chen is a former beamline scientist who just moved to Taiwan. He set up a lab down there. He's running the X-ray microscope in Taiwan. Valentina, another former research scientist, beamline scientist, who just moved to Diamond to set up the, to run the soft X-ray microscope at the synchrotron in England. Axel Ekman is our computational guru who did all of our writing the software for the automated alignment, reconstruction, and all of our analyses. And Vika was a postdoc in the lab. You can see the busy day. You can have questions if you have questions or if there's time.
And I'm sure there are many questions. Was not Chen on that bioarchives
Yes,
papers? His
yes,
first?
yes.
Is he the one that didn't quite understand when can it be done in an automated way? Because I guess he had to do it also manually for yeast.
He did it manually, and I think in reality, time became the limiting factor there. Yeah.
Understand that.
Yeah.
Right?
So that.
But if in your instrument, if you have so many wells and each one is taking what 10, 15, 20 minutes, if you do enough cells, as a part of where they are in the cell cycle.
I haven't tried that yet. I should. What I didn't point out is with that capillary, you don't just image one cell. You can go up a row of cells in the capillary. The other thing with the yeast project, again, time was a big factor there. They used a capillary that was larger than you really needed to use to image yeast, but they got more yeast in field of view, but that dropped the resolution. So it could be done with a field of view, but it would be a much better image of that particular yeast.
The other thing I'm seeing, at least I didn't notice any ribosomes. That's typically the thing that they all show when they do cryoelectric. They do the soft X-ray tomograms to get an idea where to start doing the milling. But it seems like there is a lot of complementary information that you can obtain.
Our move to Stanford will go down that pathway because Pete is Morner's former student, and his project there was correlated light and cryo. And I was part of the CZI grant that funded that project. So he got hooked on ET and SXT at the time, and he's going to be a major player when we move it to Stanford and do all of the work that it takes to make it easier to go from soft X-ray to electron tomography. So it's going to be exciting. We've got 2 fantastic people at Stanford.
Last question, was it Mark and you built the end station?
Mark built the whole thing. That whole thing you saw, all of it, and everything, absolutely everything, all of the parts it takes to image a cell. Very talented physicist.
Is he your husband?
Yeah. So I'm biased. Sorry.
Oh, but hey.
But he is.
Yeah. I'm fascinated in this work. Any other comments,
And
questions?
I had to truncate what I showed. There's just so many more things.
Oh, yeah, no one could just spend days talking.
Yeah, I could.
How do we get on your list? Be able to send samples to you?
Well, we got to build it. We have to move it and rebuild it.
And when do you think, if all goes well?
In DC on Tuesday. So
Okay,
the groundwork is there. Mark is already designing the boxes to move everything.
okay.
We're just waiting for the money to hit the door.
So maybe in 2026, you're up and running.
The front end,
Mhm.
because they have limited number of engineers and you have to get in the engineers list.
Oh, boy, okay.
So
They're not federally funded, I hope.
It's all DOE thing.
Yeah, please.
Yeah. So the one thing that I'm always excited about is the 360-degree resolution. So I just don't fully understand the mechanism. So is it that you take it like you have projection,
Yep.
and so are there no low resolution spots then in an image, or does that give you uniform resolution even though you only rotating it?
So
Yeah, you go around the whole cell, and that leaves, like, even though the caps are along the axis of rotation, for example,
and
you
again,
don't lose any resolution there.
It's minimal, it's very, very minimal, and the work Axel could talk forever about.
Yeah.
Axel's fantastic, it's just getting him to write papers is the problem, and I can't write that kind of paper, it's way over my head.
That's really exciting.
Axel's done some pretty remarkable things.
No, that's fantastic.
So
I'll ask you more questions.
you made a comment about the desktops, because it's, you know, or mentioned that you'd have to do it for longer. So does that automatically increase the photo damage?
It decreases increases your issue with stability, but it should be it's the same 10 to the 8th gray. We always end up, it just takes longer to get there. At least theoretically, that will be the same. I have seen the microscope work.
You ever
Yes,
thought
but-
about building that in addition? I mean, it seems like it's great to have an Excel.
If we put in our proposal that was rejected, big part of the problem was that the synchrotron at Berkeley is undergoing upgrades and has cut our beam time 50%. So we didn't have the people, more interested people, because if you only have half the number of days of a beam in a year, you can only
have as
have
much.
half as many users. And that they didn't like. Also, it's going to go into dark period, complete shutdown for a couple of years, and they didn't like that. So we put in the proposal buying a tabletop to try and
No kidding.
mitigate that problem, and the reviewers hated that concept. How do you ever sell an instrument if it has to be tested before you can?
We know the problem so well.
Yeah, so
Anyone else?
I had a question, I think, thank you for talking about this. I had a question about the symbiont
Oh.
vision. You showed a series of
Yeah.
images, but those weren't the same.
not the same
Right,
cell,
so-
no,
Just wanted to
unfortunately,
clarify.
no, they're, after one tomogram, they're gone, nobody gets out alive.
That's not-
It's a one-way trip in there. Yeah, so it took, it was a number of different cells on a time.
That makes sense. Yeah, thank you.
Well, then I think it's approaching the hour, so let's thank Carolyn one more time. It's a really